I'm Chad Schultz. I'm a documentary filmmaker from Philadelphia, and I'm Annalise's dad. God, she loved this room. I actually gave it to her as a present for Christmas. She moved out of a small room into this bigger room, and we decorated it and repainted it exactly how she wanted. At least she got to enjoy it for a few months. As you can see, she has a mild obsession with pandas. Annalise and I were really close. We'd make each other laugh every single day. And she just had so much energy, her whole personality. She was just electric. When she started to get sick and you saw her energy just completely go away, we knew something was really wrong. She started having double vision, and then one day that perfect smile of hers went crooked. We took her to the Children's Hospital of Philadelphia, and that's when we discovered that she had a brain tumor. It was August 7th, 2019. And I just remember sitting at that table, looking across and seeing the tears in everybody's eyes as they told us that there was nothing they could do.
There are just some locations where we are unable to remove the tumor. We may be able to get tissue and do biopsies, but they're in locations that prevent us from completely removing the tumor. Many tumors we're able to completely remove, but we know with the biology that they're going to come back.
It's 2019. I just figured, technology is moving so fast. Like maybe if we could just give her some time, she could get those 1 to 2 years. Like science would catch up with her.
Happy New Year.
Whatever time we were trying to get, it wasn't going to be enough. 8 months is all the time that we had with her. She was 9 years old.
South Dakota, Tennessee, Texas.
Heartbreaking to think, like your daughter and others, that we weren't able to get it fast enough and push it through fast enough. I can't tell you. I'm so sorry. I only met her briefly, but she was absolutely beautiful and wonderful. The little bit that I got to know her.
We just miss her.
Yeah.
What drives me in my work is really the bravery of children who are diagnosed with the terminal cancer and their parents and the courage that they have. They have very limited time with a very terrible diagnosis. We have the tools and we have the knowledge to change this status quo. And this is a status quo that we cannot accept.
We need to talk about it. We need to say that children are still dying from cancer and that we want to make a difference and we want to stop that. And you can be part of that.
The pace of discovery does not begin to match what our children need. This is one of the most frustrating truths that we're all struggling with. And it's really a driver for CBTN. That word accelerate is everywhere in our mission and our vision statements because we really have to find ways to accelerate new discoveries and the testing of those discoveries to result in new therapies. We can't do this fast enough. At the core of CBTN, and the reason it's able to achieve anything, are the tissue specimens that have been donated by children with brain tumors and their families.
Soon after that, we were asked if we were going to donate her brain tissue to research through the Gift from a Child program.
The Gift from a Child program is an initiative of the Swifty Foundation that was created by the Gustafson family in honor and memory of their son, Michael, who died as a result of medulloblastoma. So at the point in time when the physician and the family have decided that they'd like to hear more about the donation process, then I'll have a conversation with the family so that they understand they'll have all the time that they need with the child, that all the arrangements are made, and we don't have to bother them further at the time when they're the grieving of their child.
Once the surgery calls and we know that the tissue is in, every minute basically counts. We want to be right on site to make sure that the tissue is properly banked, and part of that tissue actually ends up in the laboratory, where our team is quickly working on making sure that we can generate the proper models from that fragment of tumor.
We're giving them an opportunity to take something back that was taken away from them. They may not be able, obviously, to help their own child, but at some point in time in the future, in the name of their child, they are helping other families.
If doing this could save another family from having to go through this nightmare, then yeah, we're going to do that. She's going to do that. And that was the day that we joined the Children's Brain Tumor Network.
You know, the Children's Brain Tumor Network, or the CBTN, really began as an idea. And like all ideas, it changes across time. To be a scientist that can actually affect people's lives in the present through partnerships with other amazing scientists and clinicians changed for me a model of how research is done.
Since I started, which was by design, to partner with a scientist, Adam Resnick, who we met when I was a resident in neurosurgery and he was a first-year graduate student. Having someone who would focus on surgery and patient care, partnering with someone who is a full-time scientist who shared in our philosophy about research, sharing, trying to accelerate discoveries, and that partnership for us really worked and made sense. Everything we did was not only thinking of that particular patient or that particular specimen, but how could we do it in such a way that everyone could benefit from what we were doing, not just us or our institution, but it was really a patient-centric endeavor that we started.
First, really focusing on creating a foundational layer of biospecimens that are attached to longitudinal clinical data.
10 years ago, if you would want to do research on a particular tumor, tissue was really fragmented, data were really fragmented, and it was almost impossible to capture a good number of specimens to work with. What CBTN has really allowed us is that it has brought the resources from many, many institutions into one place. They have empowered research, which is one of the most important contributions of CBTN.
Even though they were sending samples here to Philadelphia at CHOP, but the repository and associated resources were equally theirs. It didn't belong to CHOP in any way. It belonged to the community.
Right, they're sending their samples here. And so there's that, you have to get over that sense of feeling of loss of control, which I think has been such an impediment to sharing in the past, that, you know, we all know how precious these samples are, and that to then be able to really have the trust that when you're sending them to this common repository, that you're actually empowering them. You're gaining more control.
On top of that has been to empower that data in a highly connected, cloud-based environment that is queryable and is available for discovery.
Many people in the field, particularly what I'm familiar with in pediatric oncology, absolutely are willing to share openly. It's not the willingness, it's the actual doing the work to make it work and make us be able to read each other's lines of data or big genome data. It's really easier said than done, and CBTN has been the group that's trying to do it, to make these connections and allow us to share data with the right kinds of tools.
My lab, in collaboration with another lab at VCOM, recently published a study where we looked at markers in the DNA that could potentially identify children who are at risk of forming the pediatric brain tumor medulloblastoma. In the course of doing this project, we wondered if these markers were present in other pediatric brain tumors. Because of all the data present in the CBTN data repository, we were able to query hundreds of patient samples to find out if these markers were present in other pediatric brain tumors. And in fact, there was some overlap. That has now formed the basis of an upcoming project that will be starting soon.
GPC2 was identified by one of my colleagues as a good potential target in other tumor types, pediatric cancer tumor types, but not in the brain. By using the CBTN data, I was able to understand the landscape of GPC2 much, much faster than if I was trying to do this on my own and trying to collate a list of all the different pediatric brain tumors and their RNA sequencing data. But all of that was immediately available to me through the CBTN. And so I was able to use a type of immunotherapy where I take patients' T cells and reprogram them to go after a particular target, here using GPC2, was able to accelerate that work much, much faster than I would have been able to on my own by using the cell lines and the mouse models that were also available through the CBTN. And this work has shown that we can go after GPC2 in pediatric brain tumors. It's a great target and is now moving forward towards clinical trials.
And the next wave is really bringing that together on behalf of an immediate application for the patient who comes through the front door of any hospital system.
I would never get the information I need just at my center, or even at my center and a few of my friends and a couple of the biggest brain tumor centers in the world would never have enough information. For some pediatric tumors, and they're all rare tumors, they're really so rare where the information I want is all of the information in the world collected together, so I can say, based on kids that went in the past, this is the best therapy for your tumor and for kids that are the most like you. The potential of CBTN to connect the clinical data and the biology data from all of these kids who've already had to face the brain tumor is that we not lose one piece of that data for the kids who come next. I really don't think anyone should plan a pediatric brain tumor trial without requesting information from CBTN. And I don't mean it as a barrier, I just mean the data's there. You could potentially take what would have been a 10-year experiment in a clinical trial and make it a 10-minute experiment and waste absolutely not another minute before we can use that information to help kids who are going to be diagnosed with a brain tumor next.
Hello, my name is Jace Ward and I'm 22 years old. I've been battling DIPG for the last 2 years of my life. My CAR T-cell trial would not have been possible if it weren't for cell lines that were in the Stanford lab for the last 3 years. It's bittersweet for me knowing possibly one of the cell lines came from Carson Hall, who grew up just 10 miles away from me. So for me, for all the other kids out there with tumors, the time is now. We must push research out of labs and into trials. We must all have the same understanding that Carson's family had, that sometimes we have to make sacrifices. Fun, data-inspired research now.
We know a lot more about many of these tumors. The high-grade tumors, the DIPGs, medulloblastomas. It's really time to develop clinical trials that are heavily biology-based and are heavily biology smart.
I think the next decade or decade and a half, we're just going to see some amazing advances. Just the network itself, and really being able to bring this many institutions and this many researchers together for a common cause. I think that maybe is the biggest breakthrough, is that we've been able to get so many talented people coming together.
It's good humans doing good things for good reasons every single day.
It's an amazing partnership with the families that entrust their children not just to me and CHOP, but to the entire CBTN.
Almost the entire 10-year start of the effort, it was entirely funded by foundations and families who believed in this new model of working.
My son David was diagnosed at 16. David only survived for 20 months. I use the word magnanimous a lot about David because he just had this outlook on life that we should all be able to get along and we should all be like the very best versions of ourselves. Once we got involved with CBTN, we were incredibly proud of the way data was being shared, not only among members of that consortia, but really with researchers around the world. And it was not very long that I asked for David's tissue to be sent from a hospital that was not part of the consortia, and have it sent into the biorepository for CBTN. And I just remember thinking, like, this is it, right? This is what we were trying to do. We were trying to make sure that families could know that their child's legacy would mean something, and that they would be a part of something larger than themselves, and that would be a perpetual resource. It wouldn't go away, and it wouldn't just sit in a lab and not be used, that this would be used by as many people as possible. And sorry, I tear up a little bit on that, because I know that's what my son would want.
From time to time, we have an interest from family to see what we do. We're able to show biorepository, our daily operations, the models we develop. And it's amazing how surprised they are seeing those cell lines on the screen, and just knowing that one of these cell lines is with their own child tumor. It's also so rewarding how much it means for them what we do.
These are actual living cells from Annalise's tumor, which means a tiny part of her is still alive, contributing to the cause to find a cure. Often throughout the making of this video, I ask myself, why am I doing this? Going through all this pain and, reliving, some of the worst moments of my life. And it's because of these kids who are fighting this now. It's about the families that are going to be told next month that their child has a brain tumor. These kids, they gave their lives, they gave their tissue. We owe it to them. We owe it to their parents, to everyone who's donated. Annalise, she did her part. She donated her tissue. She's part of this movement now to cure brain cancer, and so am I, and now so are you. Please, if you can, support the CBTN. If you're a researcher, join the network. Get involved. Share your data. If you're the parent of a child who's recently diagnosed, I am so sorry that you have to go through this. But just know that the CBTN will not stop.
We lost a child this week who was playing from the IPG, who was playing soccer the day before. They don't have time. There's no warning for them. Families find this terrible tumor, changes their lives forever. Some choose not to be involved. You have chosen to be involved and to help us. So I really thank you for being here. It really takes a community. It really takes different disciplines to push this and to change this status quo, which is horrible. And we will not stop.
We won't 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 stop.
I won't stop.
We 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 won't stop.
We
won't
stop.
The Children's Brain Tumor Network. Until every child is cured. Brain and CNS tumors are the most common cause of disease-related death in children. The CBTN needs your support to accelerate research, drive collaborations, and launch new trials. Together, we can give more children more options, more hope, and more time. You can support the CBTN by sharing this video and by making a contribution at cbtn.org.